Well, good morning, everyone. Thanks again for joining us for our monthly EMX, normally the first Monday of the month, and the last one for 2021. If you remember, a year or so ago, we were expecting normality to resume this year, but it hasn't. So let's hope it really does next year in the future. And just a quick announcement is that next month's meeting, because the proximity of the new year, will be on January the 10th, not January the 3rd. So the second Monday of January, and then we'll resume as normal following that. And we have our two speakers lined up. But today, we have, again, two really exciting speakers. Yifan Cheng from University of California, San Francisco, and Patricia Kooyman, who is our first speaker from the Southern Hemisphere, who has kindly joined us today with Table Mountain in the background. And we hope that the Omicron, which seems to have spread first in Southern African continent, will soon be captured and overcome. But greetings to everyone. I'm going to pass it over to Wa to give the introduction of Yifan. Wa, please.
Good. Thank you, Bob. Good morning, everyone. Welcome to our EMX Symposium of the month. And it's my great privilege to introduce Professor Yifan Cheng from UC San Francisco. I knew Yifan for a long time, and he started his training in physics under an eminent electron diffractionist, Professor Feng Hua Li from Chinese Academy of Sciences in Beijing. And since then, he have traveled around the globe to pick up different techniques. He was in Oslo and in Germany and Florida State with Ken Taylor and Yoshi Fujiyoshi in Kyoto. So he actually have acquired multiple aspects of electron microscopy from material science and biological materials. And then from Kyoto, he moved to Harvard with Tom Watts, and then he settled down in the Bay Area since 2006 as a faculty member. And since then, he has been excelling in the area of single particle electron cryo-microscopy, and particularly his sensational work a few years ago to first use these direct electron detector really revolutionized the whole cryo-EM field, and his paper with David Julius, who actually getting a Nobel Prize this month, really transformed the entire field that cryo-EM become a reality. And he got numerous recognition and award. He was elected to the National Academy of Sciences a couple of years ago. And currently he has a membership of the Harvard Hughes Medical Institute. So without further ado, I'd like to welcome Yifan to give the talk this morning. Yifan, thank you for coming.
Thank you, Wal, for the very kind introduction. Let me start to share my screen. And after also apologize, I was just telling Wal that I was kicked out of my office because of my office just sounded like a fire alarm in the building. So I have to borrow a place in the neighboring building. So I hope this is going to fine. And I also want again thank Wal for the invitation to this very prestigious seminar series. So today I'm going to talk about some of our work related to the single particle cryo-EM. And I usually gave the title from atomic structure to mechanism. And so, but for this one, actually, I'm going to mostly focus on the TRPV1. And then really to from atomic structure to the gating mechanism and beyond. So just to start with looking back to the single particle cryo-EM, when Richard Feynman said that it is easy to address all the biological question, you just look at the scenes. And that actually look at the scenes was very difficult because over years that many people, particularly these pioneers, had made a groundbreaking work to establish this technology, including from Bob Glaser, Ken Taylor, demonstrating that a frozen hydration is the way to preserve a biologic sample, to Richard Henderson, using 2D crystal to study the purple membrane structure, And Joachim Frank established the single particle image analysis, and also determined the first structure, also is low resolution of a ribosome. And then also Jack Dubischer and invented this frozen hydrated technology. Those are all this fundamental sort of milestone work really established the foundation of what we enjoy and call today single particle cryo-EM. And a major part of my research has been trying to also to advance the technology of single particle cryo-EM, try to improve the resolution. And in my own lab since 2006, I've been really focused on how to improve the resolution by various ways. By the end of 2010, the field of a single particle cryo-EM was really like in the status, like in the hardware level, we are using the broadly using the electron microscope with a field emission gun, but most time we're using still side entry holder, but started to have a Titan Krios. And then for the camera, it's still the scintillator-based camera for the digital camera, CCD or CMOS. But most people still recording image with Kodak film, that's SO163 and followed by digitization. Software part, at that time, still many different packages, like including Spider from Joachim Frank and iMagic from Martin von Hill and EMAN from Wachu and Steve Lutka and Freeline Liko Grigov, just to name a few. And automation was already in place, but was not broadly used. Around that time, I remember 2008, we got in this structure of a Akira 20S proton to the resolution about 5 angstrom. That was a really good resolution at that time, but still not yet towards the atomic resolution. And a major sort of breakthrough was actually 2010 when Hongzhou's lab and then determined the first structure of this acquisition virus to the 3 angstrom resolution and then able to build a de novo model. That was really the first time demonstrating that it is possible to build a de novo atomic model on the protein that is unknown. Also, this case that the virus is an acquisition virus, very large with high symmetry, and had a certain way of so this way was still found was difficult for general protein. So real breakthrough particularly in the hardware came around like 2012, and then that was really the introduction of the direct electron counting camera. In particular, I would really want to mention that the single electron counting camera, this was really a revolutionary among the a number of versions of a direct detection camera. And this one that really a major part of work was actually pushed by David Agar and my colleague at the UCSF. And then the single electron counting not only really like can centroid in the pixel and reduce the point spread function to a single pixel, but importantly, they can also really remove the Landau noise. That is the energy of electron when deposited to the sensor that generating different energy. And by counting single electron that can remove this Landau noise. And this really significantly improved the DQE. And nowadays, this is almost every camera still using or using the single electron counting. Back then, the K2 camera from Katang was the only one that has this capability. So that's why we think it's really revolutionary. And then with the K2 camera, and then we went back to look at the frozen hydrated Akira 20S proteasome. This was the first image. This is one of our first image. The results was really stunning, that is showing the tone ring towards the 3 angstrom level resolution. That was very exciting. And then from there, we're hoping to really improve the resolution. But the introducing of this camera, because of the high frame rate, it also enabled another thing, that is to deal with electron beam induced image motion or specimen motion. This was actually first explored by Niko Grigoriev. And showing here is a paper that published in 2012. There was the first one demonstrating how to use in the movie to movie mode to actually correct for the motion. And on the left, this is the image. It's simply by averaging the stack of movie frames. On the right, this is an image that after the individual virus particle being aligned with each other. And then you can see that the streaks are all entirely removed. This was actually very, very exciting. And then also this initially demonstrated that this works, but the first effort was really from the virus. So our lab also started engaging this area, and then we developed a software motion core and then to generally correct the image motion, beam-induced motion. This doesn't really care what kind of sample you're looking at it, but just simply by using this algorithm that we can record correct the motion. You can see on the left is a Fourier transform of an image with a severe cutoff, and after motion correction, you can see this tone is all restored towards the 3 angstrom resolution level. Around the same time, and then short stress also using this a ribosome particle, they were able to track individual particle motion and showing that the motion is actually local, not uniformly globally, but it is in a local region has a difference. And then also follow on this one that are based on also initial work from Grigorov lab showing that motion is also influenced by the dooming of the sample being deformed along the Z direction. And Shenzhen in our place that combining this initial approach and developed a new algorithm. Basically, they divided this image into different patches, and for each patch, he able to track the motion. Then using all this information from these patches, and he can fit into this polynomial function this way to correct for entire motion for every at the pixel level. And this one corrected also global and local motion all together. And this is also a number of algorithm that this one implemented in Motion Core 2 is now still widely used by people. So this direct electron counting camera and the correlated related motion correction really was a major push for the field of single particle cryo-EM. And in the software side, and then also a major breakthrough was the introduction of the maximum likelihood approach. That is really transition from the traditional deterministic approach to the probabilistic approach, and in determining the image refinement. And this was first proposed by Fred Sigurd around like 2000. And then really made it perfect by Shores and implemented in the program rely on that. It's a pipeline software that everybody's still using nowadays. So around that time that really the single particle cryo-EM, really the resolution was really significantly improved. Around the same time that Shores published this structure of a ribosome structure towards the near atomic resolution, and we improved the resolution of a proteasome, Archaea 20th proteasome, towards also 3 angstrom resolution. All these things able to build atomic model. However, at that time, there's all this progress also for us that are working in the cryo-EM field was incredibly exciting. But to the broad structural biology field, that the people generally think that all this structure done by EM are already something that which actually crystallographer already determined the structure. The basically already a lot of information is already known. So in a way that they would like to see something that which EM cannot be can determine a structure that cannot be determined by X-ray crystallography. So this is a place that I'm going to switch gears and then start talking about the trip channel. And the trip channel is a large ion channel superfamily. It's second to potassium channel. And then in the mammalian trip channel, that contains these 6 subfamilies. And around 1997, and David Julius' lab and identified a number of them, which is as a nociceptor. And particularly in the trip V1, there was identified as a heat-activated channel, which can sensing the noxious heat and generating the pain. And the trip A1 is detecting this electric irritants. And also independently, independently that David Julius and Adam Patapudin, they also discovered the trip M8 as a cold-activated channel that is also a nociceptor. So today I'm going to focus on the trip V1. The reason is that trip V1 is really among all the trip channels. Trip V1 is really the post-child that enjoys a very rich pharmacological tools that we can use it to dissect the mechanism. That's why we're going to focus on the TRPV1 at this talk. So TRPV1 is largely expressed in the sensory neuron terminal, and it's a tetrameric ion channel. Upon activation, it allowed influx of cation, and then particularly calcium. The influx of calcium will change the membrane, or depolarize the membrane, and generating this action potential, which sends through the neural membrane and then through the spinal cord and then to the brain to generate a pain sensation. And also this increase of calcium can also trigger the release of some neuropeptides such as substance P, which triggers the inflammation on generating the inflammatory pain. And then for all the TRP channel, that also is very diverse in different subfamilies, very diverse in the soluble domain, but in the transmembrane domain, they are actually very conserved. In the transmembrane domain, they have this typical architecture of a voltage-gated ion channel with 6 transmembrane helices that can be divided roughly into the first 4 helices in the voltage-gated potassium channel is called a voltage sensing domain, because this one sensing the voltage across the membrane. For TRP channel, most of TRP channel, particularly TRPV1, there's no voltage sensitivity. So this domain is called a voltage sensing like domain, VSLD. And then the S5, S6, and plus this pole helices forms the pole domain. And then also, TRP channel is a long-selective cation channel. It's mostly permeable for calcium and sodium, but it has a preference for calcium. But it can also is also permeable for very large organic cation, such as UPRO and NMDG, this kind of thing. So in additional, TRPV1 is what we call a polymodal signal integrator. That is that the channel can be activated by multiple different stimuli, and then exogenously, including the heat. The heat can activate channel, but it can also activate by the natural compounds such as capsaicin from chili pepper or resoniferen toxin or RTX. This is a plant toxin, and also a spider toxin, which is a peptide toxin. These are acting from exogenous stimuli. And then in addition, that this channel can also be activated or modulated, potentiated by endogenous stimuli, such as extracellular proton, which is caused by tissue acidosis, such as wound or inflammation that generating increase or reduce the extracellular pH, and then that generating the and also potential channel. And the intracellularly, they also have it can be modulated or activated by these endogenous lipids such as endocannabinoid lipids. So TRP channel has a large family that since its discovered from the David Julius lab in 1997, it has been an intensive target for structural study, but by around like 2013, there was no structure of any member of this family. This structure is entirely unknown. This is very unusual compared with other large ion channel family. And so this is has been intensively studied by many labs around the world, particularly crystallography lab, all trying to taking a crystallographic approach. So we started working with David Julius lab around like 2010 or 2009, and tried to using single particle cryo-EM to get in the structure of this channel. Our initially tried to get into a sub-nanometer resolution, but with the technology breakthrough I mentioned earlier, that we were able to, by 2013, and then getting this structure to about 3.3 angstrom resolution. Here on the left is can see that this is the image of the TRP channel embedded in the in vitro eyes, and then the tone ring, the Fourier transform shows the tone ring goes to 3 angstrom. And then this is a 2D class averages, which at that time we thought was really stunning because it started already showing this secondary structure feature in the 2D class average, which is something that we've never seen before. And then this is a density map. From here, we were able to build an atomic model of the TRP channel. And this was a big surprise to the field of structural biology, because most people, particularly the crystallographers, didn't realize that a single particle cryo-EM would be so powerful. And this work was really done together with David Julius lab, and then particularly these two very talented postdocs, Hu Zhao and Mao Fu Liao. And then the both one who is now a faculty at the University of Utah, Malfoy is a faculty at the Harvard Medical School. And then this also that the success of TRPV1 structure also really triggered people in the field that are really a lot of crystallographers start to working in using the cryo-EM as the method of choice to really for the own targeted protein. And then also another breakthrough around that, or really not, been long there for a long time, but really picking up and then really accelerating this widespread of the single particle cryo-EM was really the breakthrough in the microscopy, particularly the high throughput automation. And automation has been really pioneered by Bridget Carriger and Clint Pott. They're working for more than several decades. And around that time, that 2010, that really became very popular because this really accelerating the structure determination and make the cryo-EM into a new regime that we're able to determine structure much more rapidly or efficient than we used to do. And this is just to show you that as a first structure of the TRPV1, this is the first structure of the chip channel family around a few years that all the structure of the entire subfamily are all determined and then by single particle cryo-EM. Some of this structure, as you can see, this num-C with this shape, you can imagine this kind of thing are very or almost impossible to be crystallized. And so around like 2015, this was really celebrated by a paper that from an article, a commentary article in Nature that the title was that the revolution will not be crystallized, a new method sweeping through the structural biology. And in the background, this picture here shows the structure of our TRPV1. So that made us very, very excited and very happy about it. So now I'm going to focus on still on the TRPV1. That our initial goal is to determine the structure. And there we determine the atomic structure of TRPV1 in the able and unlikened structure. And in this one, this is on the right here. This shows the ion permeation pathway and showing that there are two restriction sites. One is the gate, lower gate, and in the intracellular side and the extracellular side, there is also restriction, which is in the location of selectivity filter of most of voltage-gated ion channels. So at that time, we also captured another structure, which is bound by the toxin. This is a spider toxin. And also this is a different toxin. This is a plant toxin. And with these two ligand bond, you can see the channel became completely open. The two restriction sites are what now became wide open. This is a full open state. This was determined back in 2013. And this two structure that completely closed the gate and then a fully open channel really gave us the two extreme example of the spectrum of this channel in response to ligand binding. And and also you can see that there's two gates in this case are both open in the complete open conformation, but there's a channel can be modulated by various type of ligand. And that's why it's a polymodal channel. And then the channel is also a signal integrator. That in the intracellular, that this is the vanillin binding site. This is a place that like capsaicin where it binds, which is near this lower gate. It can directly acting on the lower gate and try to opens the lower gate. But this is from intracellular side. From extracellular side, there's also this side region that are in the output region. This is action side by the spider toxin or by also extracellular proton. This can also open or modulate the channel. So in our work that are following this one, we really want to start understand how this all this ligand and individually or in combinatory, how they actually transition the channel from this fully closed state to a fully open state. So our first efforts is trying to look at the structure, but particularly focus on the structure protein-lipid interaction, that we will reconstitute the membrane, this TRPV1 into lipid nanodisk. And then we enable, basically using this one as an enabled lipid nanodisk in cryo-EM. This was the first structure of membrane protein reconstituted into lipid nanodisk to the high resolution. And from here, that which we actually see was that in the vanillin binding pocket here, and then in the able structure, and before because it's able structure, there's no vanillin compound actually bound here. This pocket is not empty, but occupied by a lipid density. Based on the shape of this density, we model this one as a phosphatidylate inositol. And if you look at this pocket here, that this phosphatidylinositol has a tail packed in against this pocket here. This is hydrophobic pocket, but its head group was stuck in between these two regions and then forming a very specific interaction with this residue surrounding here. Now, from early structure, we already know that when the resinifin toxin come to bind here, then this one will be replaced. You can see that when resinifin toxin bind here, that this lipid will be removed or displaced. And then this displacement causing this a salt bridge formation that this which moves that S4 S5 loop linker here, that also moves S6 linker here. And then this move of S6 helices that allows the gate to open, and then the sort of then the ion permeation pore start open, then causing influx of cation. So from here, that this work here that we concluded some mechanism of that this vanillin PI lipid that resident, we call resident lipid, because it occupies this vinyl binding pocket in the able states. Maybe its role is to stabilize the channel in a closed conformation. So upon this ligand binding, particularly vinyl compound binding, which will displace this lipid, and then that causing the channel to stabilize, being stabilized in the open conformation. So then from there, we also want to ask, because from the work I just mentioned to you, that we show that what happened when these lipids are being displaced, and by this vanillin compound, such as resinifin toxin RTX. The next question we want to ask is, how does this happen? Not only just happen, but how does it happen? So in here, that we at a low sodium concentration out of one single sample, we were able to capture multiple different snapshots of the structure. And this is by classification, we're able to identify the ligand binding pocket has a different conformation than for the density. For example, in this one, you can see that this entire PI lipid is entirely fully intact. And in some other pocket, that the tail is already been very weakened, and then or is very mobile, but the head group is still quite well shaped there. And then we have also other pocket that where this resinifin toxin start already show the density, but this density is not fully well defined. And then we call it partial engaged. And also the head group is still not completely gone, but significantly weakened. And then finally, we have this fully engaged one where you can see the RTX density was very well defined. So from this kind of distribution, we kind of range them in a presumptive way, like this way, that this gave us a suggestion that this engagement of resinifin toxin to this vinyl pocket was that it first competing off the tail, then followed by isonositide head group. And if we look at look at all the particles in every binding pocket, because this tetramer, as each one has a 4 binding pocket, we can see that there are multiple different conformation. For example, we can have a channel where there's no ligand binding yet, or we also have a pocket particle that which 2 ligand binding site is fully engaged was RTX, but the other 2 was fully still with a intact PI, but these 2 are neighboring one or in the opposite one. They have also very many different other type of combination of the ligand engagement here. And then these are the 2 density, 2 reconstruction showing corresponding to these 2 states. You can see that this red density or RTX, this blue one was a resident PI lipids. And this one, the 2 neighboring subunits are already occupied by RTX, and this one, 2 opposite subunits occupied by RTX. But furthermore, if we look at the gate region, we can see that once the RTX engage in a pocket, it will start to influence the gating of that specific subunit. And then the first movement is to move this S5 helices. And only when all the pocket was fully engaged, will move S5 sufficient large enough to move S6 helices. But looking at all this structure, we can generating a number of conclusions from here. One is that the vinyl binding site in the tetramer are all equivalent. It seems bound randomly by the by the ligand. And we could not see a apparent cooperativity. And then, but we don't really know from here that whether there is a cooperativity or not. So I just say that there's no apparent one. And then also the level of S5 helices movement is correlated tightly with the level of RTX engagement with that specific subunit. And then also the inositide head group is the one that stabilizes the closed gate only until this inositide is completely displaced, which means that the RTX is fully engaged, that this S4, S5 is not moved. And then so that's why the only fully engaged RTX move S5 sufficient large to move S6, which is lead to gate opening. And then finally, we found that we can see that we only found that when all 4 subunits are fully occupied, that the gate is open. So this is a number of conclusions that by capture the snapshots allow us to go through this process to identify identify kind of like capture that how ligand engage with this van Noort binding pocket. And then the next thing we also want to see is how does this spider toxin engage with channel? Because with single channel recording, you can see that when spider toxin engage channel, you have this long latent time until the channel is fully engaged, open. And then from there, early study that we already hypothesized that upon this spider toxin binding, this will promote the displacement of this resin lipid, and it became empty pocket. That's why it leads to the gate open. So we also want to try to capture this one. And then from here, again from one single sample, we capture multiple different substates of how channel engage. And then here that these 4 structures showing here are captured from one single sample. And then this is in comparison with the able structure where without the ligand engagement. And we assign these states as a pre-bound and pre-open, a partial open, and open structure. And there's a lot of more details about this study that which I don't have time to go through, but just relate to this lipid function. And from all this data, we can see that on here that when this channel is fully open, also we see the tail of the lipid, but the head group is significantly weakened. It's almost gone here. So this gave a further. This is first consistent with our study early that I shown you in the previously, and then this head group is the one requires to stabilize the opening, stabilize the closed gate. But here further shows that tail probably is really to providing the binding energy, and then head is the one stabilize the closed gate. Once the head is become loose, that this is probably how the gate is closed. So I will take a few more minutes, just the last few more minutes, to give you one other final aspect of this work. That's the action of extracellular proton. And then the extracellular proton, as I mentioned earlier, is related to the tissue acidosis, and particularly related to inflammatory pain. An early study already showed from Julia's lab showing that you can see that once the low pH, extracellular pH will potentiate in channel, this is really a physiology background that related to this hypersensitivity of the pain that for the temperature. And from early work of David Julia's lab, they already showed that there's identified 2 glutamic acid residue in the extracellular loop region here, and the pole loop region here. And one of them, E600, once you mutate this one, it abolishes this potentiation. Basically, this phenomenon will be abolished by this mutation. And then the other thing is that if you keep low the pH, when the load is sufficient load, that actually can activate the channel. And then this activation of the channel by low pH, extracellular pH, will be abolished by this E600, E648. So these 2 residues are critical one. So from our work here, that we show that we also then get a structure of the low pH. But here I want to give mention there is a certain caveat is that we could not only maintain the extracellular pH, we have to put the whole protein into the low pH. In the low pH, we got 3 different conformation of the one sample, and also arrange them in the 3 different one that are called ABC, different substates. If we compare this one with the neutral pH structure, we can see that the neutral pH, this E600 form a salt bridge here with this arginine residue in this vorticensine-like domain, and at pH 6, then this E600 rotates away, breaking this salt bridge, and then also release this hydrogen bonding here, so that this pole loop gradually start moving from this position towards this position, which is open conformation related to that. And this distance increasingly getting larger. The second thing is also, if we look at this tyrosine residue, and in these two positions, they are still in the position characteristically by the closed gate. And in this one, it flipped, and this one we only see in this conformation when the channel is fully open. So this is E600. And if we look at the E648, again, in the neutral pH, it forms this well-defined density from the salt bridge with a lysine residue in the neighboring subunit here. And correlated to the 6A conformation, we can see this density has these two split density, like a two alternative orotomer conformation. And then one of them is almost same as in the neutral pH. The other one is pointing away. This is 600. As you go to this 6C, you can see that this density correlated, this rotamer density correlated to this rotamer A is almost gone. So from here, we kind of conclude that this protonation is in a two-step. This is in the neutral pH. And then once in the lower pH, the first step is the protonation of E600, which release the vortex sensing like domain so that it can tilt backwards and allow this pole loop start to move. And then the second is the protonation of the E648. So these are the second one that we also reveal that the proton action is by this one. So this is the example I want to share with you today. That this is the type of the work we're trying to do, that we're basically trying to advance the methodology in cryo-EM so that our initial goal is to determine atomic structure of our target protein, in this case TRPV1, but then followed by try to address mechanistic and physiological questions. And addressing these questions, you can see that each step when you're addressing some question, then allow you to ask a deeper and a newer and a broader question, which further often demands further technology development, and which further advance the cryo-EM. And then our work is always showing as this cycle. It's like really from the atomic structure to the mechanistic understanding of how your protein works. And this work with TRPV1 was incredibly exciting for us, particularly in the a month ago, and David Juris and Adam Putin, they were also awarded by discovering these sensory channels that in TRPV1 is by the heat, and the piezo is by the general touch. So with this is really, really exciting things for us, but also that particularly for TRPV1, we still have some further unanswered question, which we will continue to work on, particularly like in the TRPV1 is a heat activated channel. So far, we've been able to show you how ligand action activated channel, but we still don't know how heat activated channel. So this is something that we continue to work on. And then also apparently using the try to capture this heat activated channel, and then may require some new methodology experimental approach try to capture this one, or computation approach. Our early work, we kind of hypothesized that this lipid here, this resin lipid plays a role in the heat activation. Upon the heat activation, that this one being displaced, lead to gate opening. But so far, I haven't been able to prove this one by capturing the structure in a heat activated channel. This is the one that we'll continue to work on. So this is everything I want to share with you today. I hope this one demonstrates that how we gone from improve the technology of cryo-EM to determine the structure and then to answer the mechanistic question. These are the acknowledgements from current and the past member of my lab, that some of them engage in this work. And particularly here, this is one I want to list all these people here. They're engaged in the, mostly engaged in the collaboration with me and the David Julius lab. They're working on various trip channels, sensory trip channel, and some other on the methodology. And also these two fellows work with Lilian Yifan lab on the NUMC, which is another trip channel. And that's the work. And finally, I want to thank you for your attention. But last, I want to just share with you one piece of personal information, which I'm very excited. Yesterday, I just finished my 42nd marathon and in the CIM, and I finished my best time, 3 hours, 12 minutes, 57 seconds. I was very happy with it. And finally, thank you for your attention. I'm happy to address any question.
Okay, Yifan, congratulations, your marathon and this fantastic structure you show us. There are numbers of questions in the Q&A. We have only a few minutes. left. And then the first question is, how big is the data you collected to do this multiple stage data analysis?
Yeah, they're reasonably large, but they are not, in each case, I don't remember the detail, but each case that each sub-states, we were able to get into was the resolution around like a 3 something. We were able to do that. And nowadays that the work actually was done about a year ago, just recently published, but the actual data was collected probably a year ago. And around that time, we were typically trying to get several thousand micrograph. But nowadays, that in our Titan Cryo setup with this fringe-free illumination, we're typically able to get in like a 7,000 micrograph per day. And then if it's 2 or 3 day session, you get 10 or 20,000 micrograph and with multi-million particles. In this particular one I shared with you, is probably less than 1 million, my guess is. I don't remember detail.
Okay, so the other question is a complicated one. Since aspartic and glutamic, all the negative charge amino acids are very important in your arguments in terms of interactions. And the question raised is, are there any tricks you may recommend to protect these carboxylic acid better than damage during data collections? So there's some in that statement, but you can.
No, we didn't have any specific trick here, because in the APO structure, there are actually the density was very well defined, particularly nowadays that for the TRPV1, we typically, anytime we're putting the sample, non-discrete quantity sample into a Titan Cruise, we were taking a whole day data collection, we were able to get about 2.2 angstrom resolution around that level, and which is a quite routine that this the side chain of this like particular E600 and C, these 2 glutamic acid density are kind of very well resolved. And I think is that for those density that when they are particularly when they form salt bridge and then this negative charge density side chain, the density is often very well resolved. And sometimes you see the density disappear early. It was mostly that when they are alone and then when they are flipped away.
Yeah, I think there are probably 2 elements. When I look at your map, I don't know what are the resolution differences in different states.
No.
So I noticed some of the same residues, you can see the density, and other states you don't, and it might have
that's right. So E600 density is often better resolved in almost all our density map, better resolved than E643. And exactly why I actually don't know, because E600 is actually more exposed to the outside. And then so I would have thought that it should be other way around, but this is apparently not the case.
No, because this is a very complicated issue, because it could to do with the flexibility, damage, or maybe a scattering difference, and also you change the pH. You know, the proton may get attached to it as well. So this is that is a really complicated issue.
Yeah,
that would you probably can do a number of analysis to dissect some of these factors to see it and not to see it.
Yes, that's a very good point.
Right. Okay. Good. There's another question with the structural determinant for the toxin. Will it be possible to design compound to displace the toxin as a potential treatment?
TRPV1, if I remember correctly, at some point was thought to be a drug target, and people try to. So there is a compound actually developed to try to acting on the TRPV1 to inhibit TRPV1, and in the clinical trial, my understanding it was that it did not progress well because that it messed up the people's a body temperature. So there is a TRPV1 is also besides this as a sensory for the nauseous heat detection, but also has other things that probably also in a way that to maintain the body temperature. Simply inhibit TRPV1 will causing that really mess up the body temperature. So that was my understanding. But certainly, the one way one can say is that whether it is possible that to block the pain
but not messing up the temperature. That is probably a one way that we know more and more about structure, and maybe that's a way to do. But so far that we haven't been focused on this one yet.
Okay, good. All right, thank you very much again, Yifan, for these fascinating updates on what you have been up to, and this is phenomenal. Thank you very much.
Thank you. And I'll be stick around and towards the end.
Right. So, good. I turn back to Professor Sinclair.
Yeah, thank you, Professor Chu. Thank you very much, Yifan. Fascinating, great work. Thank you. So next, Professor Yi Che will act as host for Professor Kooyman. So I will hand over the baton to Yi to introduce Patricia and let her tell us about her work in the Netherlands and also in South Africa. Yi.
Thank you, Bob. Well, thank Yifan for the very nice biological talk, and Wa for your moderating. Now let's move to a physical science talk by Patricia Kooyman. Let me do a brief introduction. Patricia was educated in the Netherlands, where she spent her early career, primarily in the Delft University of Technology, pioneering the in situ gas studies in the TEM. Since 2015, she has moved to University of Cape Town and holding the Saatchi Chair Nanomaterials for Catalysis. So certainly no pricing. Today's talk title is Bridging the Pressure Gap in TEM. Certainly having the pressure you can utilize is very powerful knob. Look forward to hearing about the latest. Patricia, it's all yours.
Thank you very much, Yi. Yeah, indeed, the background you see behind me is an actual picture from my balcony at home at sunset. So I'm not sitting on my balcony now because there would be too much wind noise, but I'm very close to Table Mountain at the moment. So my position here is the part of the South Africa Research Chair Initiative, and my chair is on nanomaterials for catalysis. So a lot of the work I do is preparing nanomaterials and characterizing them and using them as catalysts. So the ultimate characterization, of course, is using them as catalysts while we are actually studying them. So in transmission electron microscopy, traditionally, we work at vacuum inside the microscope. And on top of that, in at least in material science, most samples are normally prepared in an ambient laboratory, so in air, which is a problem for air-sensitive samples. And many catalysts are, in their active phase, reduced metals, which are very sensitive to exposure to air. So we want to prevent that. And at Delft, I worked in the group of Henny Zandbergen for quite a long time. And when I arrived in his group, he had just finished holders that can prevent exposure to air. So I will show an example of that in a moment. So we could study samples, what we call quasi in situ. So we prepare a material in a reactor, we put it inside a glove box, and this is an example of a glove box. This is an airtight box that is filled with dry inert, so dry nitrogen, dry argon, something like that. And it's called a glove box because here in the front, you see these two openings. It has gloves inside so that you can still manipulate your materials. So if you can find a way to put your reduced sample inside the glove box, you can put it on a TEM grid inside the glove box and then use a holder, for example, like this one, which is closed off with Yifan rings here. And you can close it in the glove box. So you put your TEM grid in this holder cup. It's just a normal 3 millimeter diameter holder cup. You close it and then it's filled with the dry inert inside the glove box, and you can transport it to the microscope. So that's how I started my career of analyzing the more challenging catalysis problems. So I'll give you a brief example. A catalyst compound that is used a lot is molybdenum or tungsten sulfide, which is often present in what's called the slab structure, where there's a sandwich of sulfur atoms which sort of form the slices of bread, and then the molybdenum or tungsten is your pastrami or cheese or lettuce, whatever you want to put on your sandwich. So this was known, but it's still not really known where the promoters cobalt and nickel are located. So we wanted to study those materials, but what we noticed when we started using this vacuum or protective atmosphere transfer is that we would have, normally you would see these slabs. So what you see here, these dark dots, are actually your columns of tungsten atoms, and then they're sandwiched with layers of sulfur. So normally you see those slabs, so that's a single slab, and here there are stacked slabs. But depending on the preparation conditions, when we used the protective atmosphere transfer, we also saw these 2 nanometer small particles next to some slabs. And again, changing the preparation parameters, we only saw these 5 angstrom small spickles, as we called them. And it turned out that as soon as you expose your sample to air, these disappear. So these are partially sulfided tungsten oxide particles, tungsten oxysulfide particles that actually disappear as soon as you expose them to air. And later, I don't have time to show you all of that now. Later, we also showed that even the slabs in their most active phase, when they're not completely sulfided, are very sensitive to exposure to air, and they will shrink over time. So that is how I got introduced to the more challenging characterization problems in catalysis. And many of you may know already that Terry Baker started on performing experiments inside the TEM in a gas cell, and Pratibha Ghai and Ed Boyce perfected this to what is called an ETEM, an environmental TEM, which is commercialized by Thermo Fisher, formerly RVI, formerly Philips, where instead of just having your normal straight column, you make all kinds of holes in the side of the column. So you add a gas cell, an E cell, an environmental cell, where you can put gas around the stage area. So your sample can be surrounded by gas, and then you add additional pumping lines here in through the pole piece, but also a little bit higher up and a little bit lower down, for 2 reasons. One, if you put a lot of gas inside your column, you will lose resolution because your gas molecules will interact with your electron beam and create a lot of noise because it's phase that moves around and that scatters a lot of your electrons. Additionally, if your gas reaches your electron gun, it will react with your electron gun, even if it's an inert, so-called inert gas like nitrogen, because of the high temperature of your electron gun. So with this commercially available ETEM system, you can work up to about 50 millibars without damaging your electron gun, But because of the long path length of the gas, a couple of centimeters, you will lose resolution if you go, definitely if you go over 5 millibars, but sometimes even earlier, depending on your exact sample and gas and everything. I had the pleasure of working with ETEMs at Haldor Topsoe Company and Arizona State University, of which I have an example here. So this is the machine I worked with at Arizona State University, and this is one of the first ones that was sold commercially. So of course here you see the normal TEM column, and then here on the side you see the whole gas system, and down here are the gas cylinders and the regulators. So if you buy one now, oops, sorry. If you buy one now, they look a little bit more boring because they've actually put all the gas pipes and linings into this box on the side here. But you can still buy something like this from Thermo Fisher. So Haldor Topsoe already published this a long time ago, which is the growth of carbon nanofibers on a nickel particle. So what you see is that the methane in this case is dissociated at the exposed surface of your nickel particle. The carbon diffuses through the nickel particle and forms the carbon layers here on the side, and it pushes away the nickel particle from even the support. And the nickel particle keeps moving as the carbon fiber grows. So if we look at some stills from that movie, you see that the nickel particle is elongated and elongated by the stress of the growing fiber. And then in the end, the nickel particle dislodges and it can reform its more spherical shape, which of course is energetically much more favorable. There's a lot going on during this growth, which of course I don't have time to go into now, but these movies were recorded at a total pressure of 2.1 millibars and methane to hydrogen ratio of 1:1 and a temperature of something like 500 degrees centigrade. So people were already working on inserting gases into the TEM. However, another big problem of the ETEM, apart from the pressure not being really close to what you would use in practice during catalysis, is that you heat your whole sample cup. And this is a sample holder. So that ring is a platinum ring, and we also use the platinum grid without a carbon film. We did experiments carburizing iron oxide to iron carbide as part of a study on Physiotrophs catalysis. I'll come back to Physiotrophs catalysis later on. So we inserted carbon monoxide, low pressure, in the microscope, and then we heated. And what you see is that actually most of the carbon monoxide reacted on the platinum heater ring. So of course our sample was also carburized because we had a huge surplus of carbon monoxide. But if you want to do really operando studies where you also analyze the products after reaction, you can't use a heater like this because more of your gas reacts on the heater than it does on your actual catalyst. So together with Henny Zandbergen and Frederik Kramer, who was one of the clean room guys at Delft, and Henny Zandbergen, of course, was the microscopy professor. He's retired now, but he still runs his own company working on designing holders. He loves designing holders. So we developed this concept of the nanoreactor. And by the time we had a working prototype, the project was expanded, and amongst others, FEI with Pleun Donau and Haldor Topsoe with Stig Helve, who was then at Haldor Topsoe, joined the team to speed up the development. But we already had a working prototype at that time. So what's the principle? A principle is that we have a closed nanoreactor. people have been also experimenting with just carbon films, and there's more and more work on graphene cells, which is really nice for liquids. But it's much more difficult to get flowing gases in that case. And we really wanted to study catalysts while they are working. And normally catalysts are working with gases or liquids flowing over them. But we will now discuss gas cell. So one of the things we did is put a resistive heater on the vacuum side of the nanoreactor. So the heater is not in direct contact with the gases. The inside of the nanoreactor is coated with either silicon nitride or silicon carbide, which are fairly inactive in most catalytic reactions. So the reactions of your gases really occur only on your catalyst. We can measure the temperature, actually also using the resistive heater, but again, that's another separate 20-30 minute talk. But the principle is that we've got electron transparent windows, we've got a closed nanoreactor, we can put the catalyst particles inside, we can flow gases over the heated catalyst, and we can study the composition of the gas phase after the gas has been in contact with the catalyst. So this is a little bit more realistic view of one of these nanoreactors, which is completely based on chip technology. That's why it was very important to have the clean room people involved in this. We started out with making a bottom die and a top die and then assembling them. But over the years, we changed the approach a bit, and we started with a single die that was etched out, which saves a lot of time because assembling the nanoreactor and aligning the windows takes enormous amounts of time. So we don't have to do that anymore with the homemade nanoreactors. So this thicker area means that you can still handle the nanoreactors fairly easily. So this is twice 2 copies of nanoreactor. The top one that seems to be floating shows the gas in and outlet, and you can see the thin part in the middle, which is transparent to light, and the bottom part shows the gas channel in the middle, this almost reddish line. That's the gas channel where everything is very thin on this side, and the 4 electrical contacts where we use 2 to run a current through the heater and 2 to measure the resistivity, which can be calibrated for the temperature. Then when we insert it in the dedicated TEM holder, you see the tip here in the center, you see the thin area, which, as I said, is transparent to light. And on the right-hand side, you see the 4 electrical contacts. So once it's in the holder, you don't see the gas in and outlet, which are at the bottom, but they're closed off with the gas lines that also run through the holder. So then when we look at a single membrane, it looks something like this. So these dark areas that you see here are the actual electron transparent windows. So this is an SEM image of half a chip, either a top or a bottom. And these pillars that you see, in first instance, we use them to align the top and the bottom chip, but you also need them to prevent the top and the bottom thin membranes to stick together. Because as soon as they stick together, the van der Waals forces are so strong that it's very difficult to separate them again without breaking them. It's like that plastic cling film that you use to cover food. As soon as it folds double on itself, it's extremely difficult to separate it again. So that's a similar principle that works here. So that's why we need these pillars to make sure that the 2 membranes don't stick together. Then this is a light microscopy image of a complete nanoreactor. And in this case, the electron transparent windows are oval. So you see the dark squares. The white spiral is in this case a platinum heater spiral. And these dark squares are actually the electron transparent areas. And you see the small pillars in the heated area, and you see the larger, wider pillars outside the heated area. Now, one big advantage of our design is that the heated area here in the center is actually a little bit wider than the gas channel. So the gas channel can be seen here where all the pillars are. And this means that all the gas that you flow through is forced over the heated catalyst. And depending on the design, we would have 41 windows. The black area here, the black square here, shows where one of the windows is located. So we would have like a spiral of 41 electron transparent windows, but you can adjust this design any way you want. Like I said, we started out with oval windows. Later, we also made spherical windows. With the clean room technology, in principle, you can make anything you want. So what does it look like in the microscope? So what you see here at the left bottom and the top right, you see two of these pillars and you see the two overlapping oval windows. And you can already see that the catalyst particles that are inside are much more clearly visible when they are located in an area where there are two overlapping windows. So this proof of principle was published in 2008. What you see here is copper metal particle, nanoparticle, on a zinc oxide support. This is a typical industrial catalyst for both the water gas shift reaction and for methanol synthesis. And because copper is extremely sensitive to oxidation, this catalyst is always first made as a precursor where you have copper oxide nanoparticles on the zinc oxide, and then you reduce the copper in situ in your catalytic reactor before you start your actual catalytic reaction. So this ultramicroscopy paper shows the reduction of the copper oxide to the copper nanoparticles. And of course, it's completed at much lower temperature than 500 degrees centigrade. But this image shows that at 500 degrees centigrade and 1.2 bars hydrogen gas, we easily get 1.8 angstrom resolution in the copper. Which brings us to a few examples. And the first one I want to show you, which was very kindly referred to by Prof. Dion in her talk a couple of sessions ago, is the oxidation of carbon monoxide, in this case on platinum, the example that I will be showing. This is one of the reactions that runs in your car exhaust catalytic converter. There are three reactions that run in those catalytic converters simultaneously on the same catalyst, but here we are only considering the oxidation of carbon monoxide. I think most people know that this is a fairly poisonous gas, and it is formed when the petrol or gasoline in your car or diesel isn't subjected to complete combustion. So if you have complete combustion, the only products you get are carbon dioxide and water. But if you have incomplete combustion, you also get some carbon monoxide, which you really want to remove because it's so poisonous. So in the laboratory and the lab scale, like the 100 milligram to 1 gram catalyst scale, oscillations had been observed. What we did is we prepared the platinum nanoparticles in the nanoreactor using the silicon nitride on the inside of the nanoreactor as the support material, so to avoid having something like aluminium oxide to blur the images. We used a large surplus of oxygen. So in principle, one oxygen molecule can turn 2 carbon monoxide molecules into CO2. So basically, we've got a ratio of 10 to 1 in terms of stoichiometry here. And we worked around 455 degrees centigrade. And this was a main part of the postdoc project of Søren Vendelbo, who worked with me and also partially with Stig Helve. So we prepared the platinum nanoparticles, as I said, using the silicon nitride windows of the nanoreactor as support material. So we loaded a platinum salt solution, like you would do when you make a commercial catalyst on aluminium oxide or silicon oxide or titania or whatever. And we dried it out simply on a hot plate in air, and then we heated it again simply in air to 250 degrees centigrade to decompose the platinum salt. So that gave us platinum nanoparticles of a wide size distribution. The particle I'm showing here, we only see part of the particle. The whole particle is about 30 nanometers in diameter. So we inserted the oxygen and the carbon monoxide, and we started heating. And then weird things started to happen. What you see here in the bottom graph is the power we need to put into the system, into the nanoreactor, to keep the temperature stable at 455 degrees centigrade. Well, if you realize that the oxidation of carbon monoxide is an exothermic reaction, so it creates heat, as soon as the input power drops, means that your reaction is going faster because more heat is generated by your reaction. And we saw these weird oscillations happening. And as you can see, in conjunction with the oscillations in the heating power, we also saw huge oscillations in the nanoparticle morphology, shape. It went from spherical to faceted, spherical to faceted. And you must remember, you must keep in mind that this nanoparticle is like 30 nanometers in diameter. So this is literally hundreds or thousands of atoms that are changing position very quickly. And in conjunction with the oscillations in the power that's generated by the reaction. So what we can do is we can revert the input power to reaction power, which is what we've done here. So this is very similar. You see here the time also indicated in the top left corner. So we see these oscillations in the reaction power generated by the reaction in conjunction with these huge oscillations in the morphology of the particle. And simultaneously, that's depicted here in the bottom graph, we see that the conversion of carbon monoxide is much higher during the oscillation. And when the oscillation stops, we see that the conversion of carbon monoxide goes back to a sort of more steady state level. You don't see the oscillations in the gas composition because the mass spectrometer, the residual gas analyzer, is probably about 1.5 meters away from the outlet of the nanoreactor. So the gases have 1.5 meters of gas piping to mix a little bit. That's why you don't see the oscillations in the gas phase at the same speed as the oscillations in the heating power and the oscillations in the nanoparticles. What you must keep in mind is that it's not just one nanoparticle inside the nanoreactor. So all these nanoparticles must oscillate synchronously in order for this heating power to oscillate like that. So some other people at Tropes did quite a lot of Dft calculations. We're still not sure what causes these oscillations. They seem to be fairly spontaneous. We can trigger them by increasing the temperature by about 0.5 degree or 1 degree. But they also, if we just wait, they occur spontaneously and they peter out spontaneously. And we're not sure what causes that, unfortunately. At first, of course, we thought that something was wrong with the nanoreactor. So when we'd seen this a couple of times, we just loaded a new nanoreactor, started from scratch and did the same experiments the next day with a fresh nanoreactor. And we saw exactly the same thing. So the experiments that I'm showing here were performed at the Tropes facility. Then we took those nanoreactors to Delft with a duplicate holder, so not even the same TEM holder, but a duplicate holder, duplicate gas system. And again, we could reproduce these oscillations. So this is something that I would love to still spend some more time on, but unfortunately here in South Africa, exhaust gas catalysis does not have the highest priority. But there are lots of other interesting reactions that South Africa is interested in. And one of them is the Fischer-Trops reaction, which will be my last example for this presentation. So I mentioned iron carbide. The other main catalyst for Fischer-Trops catalysis is cobalt. So together with the group of Anja Sjastad in Oslo, we prepared cobalt metal nanoparticles using the hot injection burst nucleation synthesis that they did a lot of work on in Oslo. So we used a surfactant. We injected a cobalt carbonyl precursor, which then decomposes and forms cobalt nanoparticles, which are covered with some oleic acid, which protects them. And we can then load these nanoparticles again inside a nanoreactor and again use the silicon nitride windows as a support material. Now, for those of you who are not familiar with the Fischer-Trops reaction, it's the reaction that's used to make synthetic fuels. So amongst others, the stuff that is sold as biodiesel at petrol stations is made using the Fischer-Trops reaction. So what happens is you take what's called synthesis gas, which is a mixture of carbon monoxide and hydrogen. And depending on the ratio of hydrogen and carbon monoxide, you can use different catalysts. But again, that would go too far for this presentation. But the essence is that you take carbon monoxide and hydrogen and you couple the carbon atoms with some of the hydrogen into hydrocarbons, which you can use as transportation fuel and jet fuel. And you produce 1 molecule of water for each molecule of carbon monoxide that you react. So actually, this is a very efficient way of making water with a little bit of byproduct of transportation fuel. This reaction was pioneered by the Germans, Fischer and Tropes. That's why it's called the Fischer-Trops reaction. And the picture below is of Franz Fischer at work in his laboratory in 1918. And I must say, I am surprised at how airtight their equipment was, because he was using the most efficient leak detector for hydrogen and carbon monoxide that you can think of. If you have a leak and you're smoking a cigar, you will definitely blow up your laboratory. So what did we do? We tried to visualize the Fischer-Trops reaction, but the nanoreactors, although they've been tested up to 10 bar pressure difference outside the microscope, the people at Delft at that moment, Henny wasn't the sole owner, and he was definitely not the responsible person for the microscopes. So the people there wouldn't let us put in more than 1 bar gas. So unfortunately, after almost a week of struggling, we hadn't really observed the Fischer-Trops reaction taking place. So then Friday afternoon, we were like, well, let's just see what happens when we switch off the hydrogen. And there is a reaction that's actually very annoying when it happens in industry, and it does happen in industry, which is the Boudoir reaction, because Mr. Boudoir was the first one to realize what was happening. That's the disproportionation of your carbon monoxide. So you see that 2 carbon monoxide molecules react to form 1 carbon dioxide, which of course stays in the gas phase, and 1 carbon atom, which stays on your catalyst. And it's known that this reaction can lead to the formation of carbon nanofibers, and it's even been observed that huge, like 20-30 meter industrial reactors of stainless steel, thick stainless steel, burst open due to the formation of these carbon nanofibers. So as I said, we use these cobalt nanoparticles, and in this case, we ended up, after switching off the hydrogen, with 1 bar total pressure, and we had carbon monoxide and nitrogen 1-to-1, and we were working at 500 degrees centigrade. And this was part of the PhD work of Marien Bremmer, who is the first author of this Faraday discussion paper. So you see here the despacings in the starting cobalt metal nanoparticle. The time that is posted everywhere here is the minutes and seconds after switching off the hydrogen gas. So at first there was a little bit of residual hydrogen gas, but then after just over 4 minutes, we started to see the formation of these carbon layers, and they kept growing. And we saw that as soon as any of these nanoparticles was completely encapsulated in a layer of carbon, or it could be a single layer on one side and multiple layers on the other side, the reaction on that particular particle would stop. So the graphene or graphite layers prevent carbon monoxide from reaching the cobalt nanoparticles and reacting. So in a movie, the particle is at the bottom right here. You see that there's a little bit of change in contrast, which is also due to the stress that the formation of these carbon layers put on the nanoparticle. And we're not 100% sure about the mechanism, but we suspect it's similar to what we've seen with the nickel nanoparticles in that Trops movie, where the carbon monoxide in this case dissociates and the carbon either diffuses over the surface or more likely diffuses through the cobalt. Because if we look at 2 successive frames, we see that a layer of carbon is formed in one go. We don't see the gradual growth of the carbon layer. We see the complete insertion of a full carbon layer in one go. And that Marine quantified that. You can read it all up in more detail in that paper. But we really saw that we had this jump where each time a full carbon layer was added to the stack that was already present. So again, we think the carbon monoxide is dissociated on one side of the particle, diffuses through the cobalt nanoparticle, and then deposits a layer at the other side of the particle. And in that movie, we also saw twinning, and these are some stills. And the change in structure, we think on the one hand is caused by the diffusion of the carbon through the particles, but you also see a change in morphology. And we think that's caused by the stress of these carbon particles on the cobalt particle. So the carbon layers, the encapsulation of the carbon, of the cobalt nanoparticle by the carbon causes stress, which also causes some changes in the atomic arrangement. So this is only the part where we see the twinning, changing the structure. And this, all my movies, as you have seen, were looping. And so I can keep this one looping during the discussion, or I can just go out of the presentation. So that's what I would like to share with you today. Thank you very much. Should I stop sharing or?
Yeah, sure. Thank you, Patricia, for the really beautiful talk. I'm blown away by the video you recorded. And hey, let's open up for the questions. I think there's a number of them flowing in. Patricia, let me start by asking from this beautiful membrane design. I think this from the audience, there are a couple questions. First of all, what's that membrane materials, right? They are transparent to the E-beam. And second is how thick that is.
Yeah. So these home-built nanoreactors, we mainly use silicon nitride, but we also did some experiments with silicon carbide. And silicon nitride was a little bit easier for the clean room guys at Delft, but it probably just depends on where your expertise lies. Our windows were fairly small, about 20 micrometers, the spherical ones, and ours were very thin. They're about 1.5 nanometers thickness. So as many of you know, there are several companies that sell similar systems, but their windows are usually bigger and a lot thicker. So their windows are often around 50 nanometers in thickness, which means that you insert a lot more noise in the images. So with the thinner windows, you get higher quality images, but you have to be a little bit more careful because they are more fragile, of course. The thicker windows are more solid. But there's always pros and cons of everything, of course.
So Patricia, another question from the audience. This beautiful catalytic reaction, right? You know, one question people like to ask is, well, what about the beam-induced effect? As our chair right here, Bob Sinclair's beautiful study, I remember the Eileen and you know, really looking at the decomposition. So the question is, without a catalyst, you know, as a control, just seeing the beam effect. So I think the audience wants to know.
Yeah, well, it's difficult to really determine the beam effects during the reaction because you can only study the particle that you're actually beaming. If you're not beaming it, you can't study it. But we're beaming only one particle. And from the analysis of the gas phase, we can see what happens with all the particles that are present. Mm-hmm. And like I said, for the platinum, the oscillating effects, It can't have been only that one particle, because if it was one particle that's oscillating, you can't see that effect in the power of the whole nanoreactor. So it means that all particles must be oscillating in conjunction even, because otherwise the effect of the higher reaction heat would level out. And like the gas phase was leveling out because of the transport to the mass spectrometer, you would have seen not an oscillation, but just a jump if not all particles were oscillating in conjunction. So that's how we conclude that the
speaking of that, right? It's really interesting you mentioned that. I think this is related, you come back to oscillation one. Would you be able to zoom out a little bit? So you can see like 2 particles, 3 particles oscillating at the same time by observation. Would you be able to see that yet?
We haven't tried that. It's very difficult to optimize the loading of the nanoreactor. So for the very small particles, like the 5 nanometer particles, you don't observe the oscillation, but I don't know if that means it's not happening or if it's just too small to observe it easily. We saw, like I said, we saw the same thing in 2 different nanoreactors in 2 different microscopes. So we are sure that it is reproducible, but we haven't tried to zoom out to try to catch multiple particles at the same time. For the other experiment that I showed, the cobalt nanoparticles that are being encapsulated in carbon layers, there it's fairly easy to afterwards study many other particles that had not seen the electron beam. And there we saw that there was a variety of particles in the amount of layers of carbon that they had. So we saw some of them that had only 1 carbon layer, and we saw quite a few, like the one that I showed that we were imaging during the reaction, that had multiple carbon layers on one side, and then either none or a full carbon layer on the other side. So that's how we concluded that as soon as the particle is completely encapsulated, the reaction stops. So we saw particles with many different amounts of carbon layers after the reaction, because once we cooled down and taken out the gases, we could just image as many particles as we wanted to and see if they were all similar to the one that we had been imaging during the reaction. So yes, they were similar, definitely. And it wasn't the case that the one that we imaged during the reaction was the only one that had like 8 layers. There were more that had 8 or 10 layers, but there were also some that had only 1 layer or 2 layers.
Yeah. So Patricia, for the time consideration, let me ask the one last question. So you're showing this oscillation reaction power oscillate, and then the shape changing facet, round shape facet. So when the reaction power is high, is it I couldn't tell right away with my eye, right? Is the round shape or the facet shape? Which one that is?
Oh, I would have to go back to the paper. Sorry, I don't remember off the top of my head.
Okay. Yeah, that seems to be very interesting, and could be important also, because when the reaction power is high, right? You know, the surface energy for the facet and the round shape will be different.
What we think also from the DFT is that the when the reaction power is high, is actually during the change. Because what happens is that when it's faceted, the surface is completely covered with carbon monoxide. And then somehow 1 carbon monoxide molecule reacts or leaves, and then you suddenly get a change to the more rounded particle, which is completely covered in oxygen. So the fast reaction actually takes place during the change, because when it's faceted, it's completely covered in carbon monoxide. When it's more spherical, it's completely covered in oxygen. So the reaction takes place during the change.
Very interesting. With that, I think, Bob, I will come back to you.
Okay. Well, thank you, Patricia. That's for a very carefully explained talk about really how difficult it is. And I think one of the main drivers of electron microscopy in the 1930s was the attempts to do in situ work in gases and in liquids for improving the resolution over the optical microscope. So this is an equally important development over the years, and it takes many years to get to this stage. So thank you for your talk. And also Yifan for his brilliant talk as well. So we will stay online for people who'd like to continue the discussion with our speakers. I don't know if Wa may want to add a few words, but I'd like to send everyone best wishes for the upcoming holiday season, and to remind people that we will start again, not on January the 3rd, but on January the 10th, with our next speakers, David Wiesler from University of Washington and Zhao Ching Pan from University of California, Irvine. Wa, would you like to say something?
Yeah, well, I think this is a phenomenal morning to start with this week, and I really enjoy both Yifan and Patricia's lectures. That was a good treat at the beginning of the week. And I am trying to try some new avenue for question and answers. If you type your questions in the Q&A, I would let you talk to address a question, so we can try a dialogue between the speakers and the participant. Could we try that? If you just type in your questions. Okay, why people typing in? Let me start with Yifan. Yifan, so since you have been really heavily engaged in the channel structural mechanisms, what do you think will be a limiting factor in trying to tackle this problem? If an investigator have another channel, they may not be as big or as well behaved as the trip channel. And so can you just make some comment from the beginning of the biochemistry to the validation or the resolutions?
Doing structure is one thing that certainly there is a very, from protein purification expression and purification until you do the structure. And then by the end of the day, you're judging that whether you get a high resolution structure of all the channel, of every part of the channel. And so it seems to me currently that really the real challenge is still a lot of them lies in the biochemistry. And then once you have a very behaved protein, oftentimes that doing structure is reasonably feasible. But there are cases, for example, in the TRPM8. So far that we spent many years trying to get a structure of TRPM8 because that we can get a whole channel, but except that the pore region, in most cases, is just simply missing. And it's hard to understand why, and it's probably still a combination of purification and all the other things that this part is not stable. And then one way people typically do is that do an Osteologic screening to look at different species and try to identify one that eventually can lead to high resolution. This is what actually we did for the TRPM8, but this is a very tedious process. And so a more probably better way is that I was thinking is that if one can have the protein, make sure the protein is functional if you purify them and that can be put into the lipid nanodisk, particularly not nanodisk like liposome or things like that, and then make sure they are functional, then that would be a lot easier. Like in the TRPV1 case, even we spent like several years engaged with the channel that from 2009 that actually for David Julius that even longer, we also spent many years working on this protein.
and then they're basically able to purify them and then reconstitute them into liposome, and then did all the patch chem on the liposome, make sure that proteins function in liposome. That's why we use the exactly same lipid composition for that liposome to the nanodisk reconstitution and able to do the structure. But for TRPM8, for example, so far we haven't been able to do that. Reason is that the protein can only be purified by detergent like LMNG, which is incompatible for reconstitution. So, which is still a bit challenging for that. So that's the part of the thing. The other part, I think, for TRPV1, particularly that which is powerful, is that there are so many known ligands that can capture the protein in like antagonist or agonist capture protein in different conformation. So it's all these tools are readily available. For some other channels that they may not be that available. So one have to think about whether then really it's not just simply doing the structure. I think it's really in combination with the physiology to understand that which way can you get the tools or you need it to trap this protein in certain conformation that you can do the structure. That is still very, very challenging. Yeah.
Okay. So I think in other words, we still do the wet lab.
Yes, I feel like the wet lab is still a major part of the our daily business. Yeah.
Okay, good. All right, thanks.
Just like you need a reactor to check that you're looking at the right thing.
Yeah.
And
Okay, Nestor has his hand now.
Yeah, Nestor's joined us. Thank you, Nestor, please.
Hey, great talk, everybody. Pat, I got a relatively simple question, I think, for you. I've been using a lot of silicon nitride films for both liquid cell and other stuff. And the biggest problem I have with silicon nitride is it's not silicon nitride. There's always oxygen, and the way they tend to make it, there's a lot of chlorine in, well, a lot means, it depends. There's also chlorine present. Have you seen any of that in any of your nanoreactor silicon nitride? Because I imagine it would influence the reactions.
Most of the reactions I've studied so far were actually in, well, not for the platinum, that was in oxidizing atmosphere. If you're working in reducing atmosphere, you probably won't get too much silicon oxide. Maybe on the outside, because of exposure to air, but the inside should not have too much silicon oxide. We actually try to make silicon oxide membranes as well, but the thing is that those are much more sensitive to electron beam damage. So once you have more silicon oxide than silicon nitride in your windows, you start burning holes in your windows.
Yeah. Okay. Well, what I see is this oxygen and chlorine is actually distributed through the window. It doesn't matter how thick the window is. It's not just on the surface. So it's in the fabrication process. And I always worry that it's going to influence whatever process we're studying.
Oh.
Well, you need to check your windows.
Yeah, okay. Yeah. We never analyzed them in that much detail, but I'm not sure how the oxygen is in the. I would have to look up how exactly they make the ones that we've been using. Yeah.
Or send me
but
some.
It's definitely.
I'll have a look at them in my beast. I'll let you know if there's any oxygen or chlorine.
Well, that definitely sounds interesting. Yeah. Thanks.
Okay.
Yeah.
Okay,